I have another question. Like
Yes, Vineeth.
if microhyla and polypedids are calling from a same location and in the recorded file, if both of their call has been recorded, then how to do analysis in this case?
You are saying, okay, polypedids and microhyla is calling. In the recording, you have both the recordings, correct? Both the species calling.
Yeah.
And if they are merged, you can't do anything. Okay. If the call of the microhyla and the polypedids is merged, you can't do anything. You have to look for parts of the sound where there is free or independent calls of microhyla as well as polypedids. Analyze only that part. There's no solution for this.
Okay.
Because if you try to remove the call of, let's say, polypid, it's going to remove even the frequencies of micron because it's interspersed.
Vineeth, I was asking a question. So
Yeah,
in the program, right? So we have this frequency starting, low frequency starting from below the graph and it goes up, right? So does the
exactly.
shape of
Let me
spectro,
show you that. Huh.
yeah. So does the
Let's
shape of the
zero to
spectrogram,
22.
go ahead, Vineeth.
Yeah,
So in
go ahead,
this
Anu.
yeah, in
Yeah,
this example, does the shape of the the call, does it help us to identify or at least differentiate um, type of the call, or where the frequency are low or high? Does the shape it forms in the spectrogram has to do anything?
absolutely. See here, the frequencies, the lowest frequency in this call is beginning somewhere here. See here,
Mm-hmm.
it's let's say around see here marking around 800 hertz, and it is going up to somewhere around. This is the second harmonics going up to 5 kilohertz. But the maximum energies are concentrated here. See, this is the frequency where there's maximum energy. That is close to around 1.2 kilohertz.
Okay.
Whichever is the darkest band is the
Mhm.
maximum energy containing frequencies.
Okay. And like if it forms a particular shape, does it indicate something? For example, like at least in birds, right? So there is, if a bird is doing whistling or hoot, right? The kind of
Hmm.
shape differs. So is it
See,
something?
shape, absolutely. Shapes are syllables. In case of birds, it is called syllables. But in frogs, unfortunately, I mean, fortunately, we can say, we don't have anything like. See, it's uniform. It's
Okay,
uniform. See, every
okay,
pulse.
okay.
But in case of a bird, I will open a file. Just wait. Just I'll show you. There are some files available uh, in Raven itself. One minute. There are some calls here only. I'll open one of the calls here. You'll be able to see.
Okay.
So, I don't know, not worked. Okay, chestnut threaded wobble. Let's open this. You'll be able to see it's totally different. Okay, you can see this, right?
Yes.
See, look at this. There are syllables. Okay, first syllable, second syllable. There are different types of syllables, just going up and down. This is what is the spectrogram of the call. This forms a very important data because this is how the spectrogram looks like. This is how the syllables are. It's an upward ascending syllable, ascending plus descending, ascending syllable. They'll define that in birds. Of course, I have not done any bird acoustics, but I believe that's what they do it here.
Okay. Okay. So in frogs, we won't find such kind of things, right?
No, in frogs, it's quite simple. You saw on all the calls. Even if you want later, you observe all the calls would have sent you. In frogs,
Mhm.
it's much simple. All the pulses will have the same sort of nature. Okay.
Okay. Okay.
Yeah.
Sounds good. Thank you.
Yeah. Vivek, Vivek Gautam.
Yeah, hello.
Did you understand what I said? Like if the calls are completely overlapping, let's say calls are completely overlapping, you cannot do anything, but you need to sort of find areas of your call where there is no overlap and analyze only that part of the call for individually for polypedals, individually for your microphone.
Yeah.
Yeah.
So I need to check the whole duration of the recording where
Yes.
the two
Yes.
calls are separated.
Yes. Free. Only analyze those because other calls are not useful for your analysis
Okay.
because you don't know where the call starts, ends, and even spectrograms will be overlapped. You cannot use overlapped or a sort of a chorus type of a call most of the time, unless you are doing something like a population study, okay, saying that, okay, these two species are present. But otherwise, we don't use it for individual species acoustic analysis.
Mm. Okay. Thank you.
Yeah.
Sir, if we are in a field and we get a group of frogs sitting together and calling, how do we get the proper calls, like one single call or something? Because we have multiple species in one particular area, maybe a pond or somewhere. So if we have multiple
Hmm.
species calling together, how do we record it separately?
See, I showed you, for example, here, right? When, like, let's say my rubra call itself. Okay. Just a minute. If you see this, Divya, can you see as I zoom in this already? Do you see there are two different spectrograms?
Yes.
Yeah. So now you know for sure this is one species. Okay. One minute. Play that and play this and see. So confusion.
Mm.
So the different species will sound different on the spectrogram as well as the waveform. Even the waveforms if you see, it's totally different. See, so even if they're calling at the same location, you need to look for the points in your call. Let's say I have a point here. At some point, they're all calling together. Don't choose that part. Go for a part where there are spacing between the calls. For example, maybe this call, see it's spaced well. You would be able to easily identify the species.
Okay.
For example, here you see, I think there's some sort of a noise. Let's noise. Two Ornata was calling at the same time. Okay. This Ornata was little far. Are you able to observe? Look at the spectrogram. See, two Ornata is calling together. Even Vivek, who was asking me the question, same. See, two Ornata is calling together. But look at this spectrogram, which is more dense, and this is more light. Means this individual was calling little away from our microphone, whereas this one called very close.
Okay.
Yeah, this is not useful for analysis, but it can be used for understanding what species is there. That's fine because see, this is very clear. Uh, it's rubra because but look at this particular part. This is one call actually. The in between one more individual is calling actually. You can see it's gone too long. The actual length is this much, but there's one more species or individual which is calling. So even if you hear it, you can clearly make out there are two individuals.
Okay.
So one is far, one is close. Yeah. But however, you can look for the regions where there is no overlap and easily can say Okay, there are 10 species calling, there are 5 species calling. Whichever is
Sir,
close will give you
does
better
this,
signal.
yeah,
Sorry.
does this also work with the dancing frogs? Because they always will be in the waterfall, no? Waterfall area, and we will have the water flow sound also coming in between.
Correct. So that's why you need to be smart. You need to go when the water flow is low, let's say about December, January, and record it at the stream. So to avoid as much as noise as possible. But
Okay.
however, of course, there will be stream noise, which will be at a lower frequency. It will be below the frequency range of your animal. So it should not matter for you in the spectrogram analysis. However, in the waveform, there may be some anomalies. It's fine because that's how it is. Another species is here.
Yeah, okay, sir. So if there are any
Yeah,
other
sometimes
questions,
we need
please.
to
Yeah.
take strategy. Yeah,
So, hi, Vineeth sir.
yeah.
So
Hi,
my,
Priyanka.
my question is, if a female frog is approaching a male frog, then the frequency of a male frog will intensify, right? So in that case, how can I come to a conclusion that this is the range of peak frequency of a particular species of frog? Like,
Yeah, Priyanka, for your question, we need to see every species and we need to record the calls before the female has approached and when the female has approached. If you're saying that frequency is definitely changing, which I don't know, and you also may be suspecting as far as I see. So if you're sure, if you already have the recording, it's fantastic because it's giving us a data that is, sort of a courtship type of a call there in the species. Means courtship means when the individual approaches, then they change their, as I told you yesterday, they change their, you know, some of the duration of the call or frequency, etc. Then it's very interesting. You can write a paper on that, saying that the species advertisement call is this, and the courtship call is this. As soon as the female approaches, courtship call is seen with a different frequency and different duration, different number of pulses. Yeah, it will be interesting work actually.
thanks.
Did you understand what I'm saying? So most of the time it doesn't happen, but since you asked the question, I feel you might have observed this in the field. If you feel that
Mm.
it is happening, males are changing the call as the females are approaching, then definitely you need to observe the waveform and the spectrogram and tell it quantitatively that there is a change in the spectral properties or temporal properties. Then you can 100% ascertain that there is a change. Otherwise, just by listening, you can't make out the changes.
Yeah, we have to do multiple recordings and we have to do
Yes,
analysis.
recording before the female is approaching, recording when the female is close. Then, if you find it, then it will be interesting, write up and research paper saying that this particular species in India has a courtship call.
Thank you.
Yeah.
Thanks, Vineet, for the great session. It was helpful to understand the
Thank you.
basics.
Thank you, Anup. Thank you.
Thanks.
Yeah. Welcome. Welcome, you all. I hope you had a good time. Even I had a great time interacting with you all. So, if you have any queries, feel free. My number is available with you. You can talk to me anytime. Even if you want to associate in any sort of research, you can always feel free to write down. Okay. Thank you very much.
Thank you so much, sir. It is again wonderful that you also had patience to extend the session to only discuss about all those doubts. Thank you so much. And participants also, I
Absolutely.
thanks a lot. Um,
Yeah.
I
Thank you, guys.
Yeah. So I'll be ending the meeting here. If anything is there, then you can also discuss on the group, or as sir told, you have his number already. Thank
I
you.
will also share with the participants some interesting research articles. When you have time, you can read how acoustics has been used in different amphibian research. Yeah.
Yes, sir.
Thank you all.
Yeah. Thank you, sir.
Yeah. Thank you. Good night.